Hi, so today we're going to be talking about the ABCD1 mutations and adrenoleukodystrophy. I'm Isabella, and with us we have Melanie McCleary, Tia Beaver, Jael Corridon, and David Joseph.
So to start us off, I'm going to talk about what ALD is specifically. ALD is ALD is a disease that affects the adrenal glands as well as the spinal cord and the white matter myelin of the nervous cords. The specific gene that goes with this is the ABCD1 gene, which provides instructions for producing the protein ALDP, which is in the membranes of the peroxisomes. ALDPs bring in a group of fat acids that are typically known as very long chain fatty acids, also known as VLCFAs. And they bring them into the peroxisomes where they're then broken down. When the gene mutations occur, this prevents the production of the ALDPs, which results in a shortage. And this shortage will then break down the VLCFAs, which then causes a buildup of the fatty acid chains, which also will then cause high level of fats to accumulate. And this whole accumulation is toxic to the adrenal cortex, the myelin, and will destroy the protective myelin that's coated around the nerve cells, which will in turn affect different brain functions. For inheritance, this is inheritance in a X-linked pattern. And with this, females have two X chromosomes versus males who only have one chromosome. So with the females, they have an X chromosome to offset the mutation, but since the males only have one, the gene will abnormally cause the disease.
Hi, my name is Jael, and I'll be briefly talking about the gene location map and sequence of the gene. So the gene is located on the XQ28 position on the chromosome, which is, after research done, is the tip of the chromosome. And the gene sequence is usually detected as the ABCD1 pathogenic variant. So this is the sequence of the gene, which is the ABCD1.
So I'm going to be covering some of the medically relevant phenotypes involved with adrenoleukodystrophy, ALD. So as we said before, we spoke about myelopathy, which involves the disease of the sheath around the spinal cord, and it would be commonly seen as progressive weakness and stiffness in the lower limbs, thinning hair, and in a younger age, in boys more typically, you would see adrenal insufficiency, and cerebral ALD, which is separate from the regular ALD. Can present itself as behavioral and learning deficits at a young age and could be possibly misdiagnosed as ADHD and stuff. And then as they get older, it can progress into more severe situations. And more typically, you'll notice that ALD is diagnosed less in women and is more commonly seen as a diagnosis after they hit 60 years old, because in women, there's a less likely chance of them having cerebral ALD, so it's not as noticeable. And more often than not, it's misdiagnosed as multiple sclerosis. So women tend to not be diagnosed or even tested for ALD unless they have a male in the family who's already been diagnosed with ALD in the past.
And as you can see, we have a figure 3, which explains a little bit more about the mutation on exon 1 on the ABCD1 gene. If there's a red arrow on the graph that shows the peaks, so this is like the mutation of the gene. It goes from AGT to, well, the code goes from AGT to AGA. So this is a substitution for a serine to an arginine, and that's where we kind of see the missense mutation. And the arrow represents that nucleotide change, and mostly missense SNPs are present in this gene.
And so I'm going to now be discussing the protein structure and function. So the protein ALDP is expressed by the ABCD1 gene, and it's a half-size peroxisomal ABC protein. And in humans, it has 746 amino acids. The ALDP is located in the peroxisome membrane. And peroxisomes, just as a refresher, are small sacs located within cells that are involved with processing molecules. And in a normally functioning protein, ALDP is responsible for transporting very long chain fatty acids into these peroxisomes, where then they are able to be broken down. And these very long chain fatty acids make up parts of brain membranes, including myelin. And so in figure 2, you can see the structure of our protein. And this is expressed by that ABCD1 gene. And you can see it's a dimeric protein. And in this image, it's specifically attached to oleola-CoA, as shown in gray, where that arrow is.
Hello, my name is David. And when it comes to gene therapy for X-linked adrenoleukodystrophy, developments have been made for cerebral ALD. Lenti-D is a hematopoietic stem cell gene therapy trial that is being investigated in a phase 3 clinical study for its efficacy in treating CLAD, cerebral adrenoleukodystrophy. This type of gene therapy focuses on the transduction of CD34 positive cells with a lentiviral vector into patients with ALD through intravenous injection. In addition to lentil-D, recombinant adeno-associated viral vectors have been utilized in preclinical trials with non-human primates. For the case of X-linked ALD, a group of researchers have conducted a study on the IV injection of recombinant adeno-associated serial type 9 mediated transduction of human ABCD1 gene into the mouse CNS, central nervous system, and the fibroblast of X-linked ALD patients. This study contributed to the understanding of how RAAV9 can be utilized to reduce levels of VLCFAs, the very long chain fatty acids, and promote ABCD1 gene expression in the brain, adrenal gland, and spinal cord. So as an emphasis here, animal models are important in gene therapy development. And finally, the implementation of low scoring when brain MRIs are being done to detect and diagnose for X-linked ALD. This process of low scoring can facilitate decision-making in patients for hemotopic stem cell therapy, especially for patients with a low score less than 9. So in conclusion, the complexity of ALD genotype and its relationship with the phenotype is of great interest in science because just the mutations in the ABCD1 gene can cause a wide variety of phenotypes. And this is important because there needs to be a continuation of therapeutic interventions because this will help improve the quality of life for those that have X-linked ALD.